everyone. In our ongoing series in biotechnology, we are discussing today the gene editing or genome editing tools. In this, we particularly talk about the CRISPR-Cas9 system, which is the clustered regularly interspaced short palindromic repeat, that is CRISPR, and the Cas9 protein associated with it, and making a genome editing tool, a very powerful genome editing tool. We are discussing that. We already discussed about the structure of this tool in our previous session, and now we proceed to discuss about the mechanism of its action. So, this gene editing tool CRISPR locus, we have already discussed, is basically looks just like this. It contains a set of Cas genes which will create Cas proteins, and the CRISPR loci contains the directed repeats, which are the DRs shown by blue blocks here, and it is interspersed by these spacer sequences, and these spacer sequences are variable sequences, and these are basically the memory of any exogenous DNA that has been introduced into the cell. For example, a bacteriophage, when it introduces its DNA, then a short segment of this DNA is retained as a spacer in between these directed repeats and stays at the memory as a memory. So, this is how the CRISPR-Cas loci looks like. When we look at its mechanism of action, the idea is that how these things have been created, we have talked about. Now, this CRRNA array, the CRISPR RNA array, is transcribed to give rise to the CRRNA. Now, this CRRNA will contain multiple units, and with the variable sequences what are there in here. Now, this will be then digested to give rise to, or they fragmented to give rise to some 20 base pair units, the CRISPR units which are there, and based upon this RNA which act as the guide RNA, it will now associate with Cas9, and then with the Cas9 protein, this guide RNA is going to guide it to the target DNA, which is homologous to this guide RNA, this 20 base pair nucleotide of the guide RNA, which is basically the homologous part. The rest of the portion will also, some of the direct repeat, some of the direct repeat also is introduced into these smaller subunits. Now, there are specific RNA that recognizes this each unit, and then there is RNase 3 that cleaves these subunits into individual units. So, it is transcribed as a single RNA molecule, and then this RNA is basically cleaved into these kind of subunits which are there. So, that is how it occurs, or an overview of the mechanism of action. The type 2 CRISPR system consists of the nuclease Cas9, the CRRNA array that we have discussed, that encodes the guide RNAs, and a required auxiliary trans-activating cRNA or tracer RNA that facilitates the processing of the CRRNA array into discrete units. So, this is another thing which is present in the CRRNA loci, which is the tracer RNA or a trans-activating cRNA. This is very important for cleaving the whole cRRNA array into the discrete units. This is, each cRRNA contains a 20 nucleotide guide sequence and a partial direct repeat, where the former directs the Cas9 to a 20 base pair DNA target via the Watson-Crick base pairing. Let us look at it in the diagram again. This is another diagram which explains the role of the tracer RNA, and again molecularly tells you what is happening. So, this is, for example, the target DNA which is present. Now, this target DNA homologous region is present as a spacer in the CRISPR RNA array. This is the CRISPR loci. So, there are Cas proteins, there would be Cas proteins. Now, there is a tracer RNA, this is just showing this kind of a CRISPR locus. There will be upstream some Cas genes as well, which is going to produce the Cas protein. So, please understand this carefully. These are the CRISPR RNA units, CRRNA units. You can see that there are direct repeats, and then in between there are the spacer DNA, and this spacer DNA has homology to the target DNA. The essential part is that target DNA should have a PAM motif associated with it. So, now this tracer RNA is going to produce this tracer RNA. This is going to produce the cRNA array, which contains multiple direct repeats and with containing spacer molecules. Now this tracer RNA will have some homology to the direct repeats which are present here. So it is going to go and homologously bind, complementary binding to the direct repeat region, just like it is shown in the diagram. And then RNase 3 is going to cleave just next to where this direct repeat is. So it is going to cleave these regions into short oligonucleotides region, where a 20 base pair guide RNA has come. This has come. So this is termed as the sgRNA molecule. So this tracer RNA is very important to cut these cRNA array into discrete units. Its complementary binding will cause the RNase 3 to cleave after wherever it is bound. So it will create these 20 base pair units, each containing some part of the spacer, and in that spacer there will be a scaffold as well as the homologous region and this kind of a tracer RNA region as well. Now this get associated with the Cas9 protein molecule. So this RNA gets associated with the Cas9 protein. Now Cas9 is a nuclease. Now this unit, the Cas9 and this discrete unit, when get associated, will be guided to bind to the target DNA, because this particular region contains homology to the target DNA. Again, the important is that there has to be a PAM motif. When there is a PAM motif and when there is a homology between the guide RNA and the target RNA, it is going to take the Cas9 molecule and bind to the target DNA. And then this Cas9 nuclease is going to create a cleavage, or it is going to create a double-stranded break, 3 nucleotides upstream to the PAM motif. So PAM motif is actually important for the Cas9 nuclease activity to be there. So it is going to create a double-stranded break. So this is how the CRISPR-Cas9 system causes the cleavage or creation of a double-stranded break in the targeted DNA. So I hope you understand that why it is very specific and targeted, because of these spacer DNAs, which is complementary to the protospacers in the exogenous DNA that is there. Now in the natural system, these kind of spacers are naturally lying because of some kind of an infection or exposure to the exogenous DNA previously, but we can manipulate it because we can artificially synthesize these spacer molecules depending upon making it the homologous to any target region that we want to target at. So these spacers can be designed and introduced in between these directed repeats, and then these can create these discrete units, and once it gets associated with it, it goes and direct and bind to the target depending upon what spacer DNA you have introduced into this system. So that is how it can be used in biotechnology as a genome editing tool. Naturally, these spacers are basically the exogenous DNA fragments, but we can manipulate it and introduce our interest spacer DNAs, which is complementary to our interest target DNA that we want to edit. So this is an important mechanism where the formation, the biogenesis of cRNA and then the interference takes place to abrogate the function or whatever the outcomes of the genome editing are. RNA guided nuclease function of CRISPR-Cas is reconstituted in mammalian cells through the heterologous expression of human codon optimized Cas9 and the requisite RNA components. So as I said, this natural defense mechanism can be utilized to create biotechnological systems, not only to manipulate the mammalian cells, but yeast cells, fungal cells, plant cells, all animal cells, all different cells can be manipulated by introduction of the RNA guided nucleases. So RNA guided nucleases can be synthesized, these molecules can be synthesized artificially and delivered inside the host cells, or they can be the genes for this RNA spacer, as well as the the sgRNA, as well as the Cas9 protein can be introduced, can be inserted into the vectors, and then these vectors can be introduced into the cells where they can express, form the CRISPR-Cas9 system, and do the genome editing by gene targeting. So the heterologous expression is important, so we can introduce into vectors, introduce them into cells. These are the normal procedure. Introduction into the vector will be a normal recombinant DNA technology procedure. Introduction into the specific cells would be a normal transformation procedures, whatever is routinely used. And there they can express these nucleases. Obviously, it is codon optimized Cas9 proteins, codon optimized because there is a variation of codon that is used between the prokaryotic and the eukaryotic cells. So, because Cas9 is a prokaryotic protein, the codon is optimized to make it to make it readable in the eukaryotic cells, for example, mammalian cells, and the other requisite RNA components. Furthermore, the crRNA and the tracer RNA can be fused together to create a chimeric single guide RNA. So, this sgRNA is a single guide RNA that will contain the part of our crRNA. So, crRNA containing the 20 base pair guide RNA as well as the scaffold RNA as well as the tracer RNA, because it is going to cleave that entire array unit into discrete units. Cas9 can thus be directed towards almost any target of interest in immediate vicinity of the PAM sequence by altering the 20 nucleotide guide sequence within the sgRNA. So, as I said, this 20 nucleotide guide sequence can be designed based upon our gene of interest where we want to target it, or any region of interest that we want to target it to, and that can be basically created. So, this is, for example, I am just showing that in this case, for example, Cas9 is contained by, this is a dual plasmid system where one of the plasmid is carrying the Cas9 protein and the other plasmid is carrying the Cas9 sequence, and the other plasmid is carrying the sgRNA sequence. So, both the sequences are carried by plasmids, and when they are co-transfected into the host cells, then these host cells get transformed and then they can be selected based upon the selection procedure for Cas9, and based upon the selection procedure, this is just a normal recombinant mechanism that is there. So, the recombinant DNA is created by the introduction of Cas9 sequence. Recombinant DNA is created by introducing the sgRNA sequence. So, this sgRNA, we can amplify it, we can synthesize these oligonucleotides, or we can isolate it from different fragments if you want to specify a particular target, and we just introduce it into these vectors and host specific vectors, and then just introduce it routinely, co-transfect it into the whatever cells we want to use, whether it is mammalian, animal, plant cells or whatever. So, different methods of transformation or transfection are there, and then we do the routine selection for the presence of both the plasmids. So, when both the plasmids are there and the heterologous expression occurs, then the Cas9 will be formed, sgRNA will be formed. Now, this sgRNA is going to get associate with the Cas9 protein, and it is going to target this Cas9, it is going to guide this Cas9 to the target which is complementary to what sgRNA you have introduced it into the cell. So, it is going to go there and it is going to shut off or activate or cause deletions or inversions or whatever the genome editing outcomes are. It is going to do that. So, this is how basically the heterologous expression occurs. So, what are the features of this CRISPR-Cas9 system? The cost is generally low as compared to the other nuclease systems. The ease of doing it, that it is very simple mechanism how to do. The specificity is high, and then there are multiple loci where it can be basically can be targeted to. The requirement is there has to be a PAM sequence which is required for the target sequence, and multiple genome editing can occur. So, this is a very high yielding mechanism that is there as compared to the other. So, I just show the other one as well. For example, for TALEN, the cost is again very high. Again, it is not very complex, but it is a little complex mechanism to generate these tunnels. The specificity is intermediate. The Cas9 is very high. The specificity in here is intermediate. It is highly effective and specific as far as the positives are concerned. The cons, for example, here is the requirement of the PAM sequence, which causes the limitation. Here it is very time consuming method to create the talons and all that thing. And again, multiple genome editing, there are very few models that can facilitate multiple genome editing in one go. Similarly, for zinc finger nucleases, the cost is low, but the specificity is also very low. So the pros is that it is effective and specific, but the specificity is lower as compared to the other genome editing tools. Again, it is very time consuming, and multiple, again there is very few models to do that. So with the advent of the CRISPR-Cas9 system, it just revolutionized the entire genome editing field. And nowadays CRISPR-Cas9 systems are applied, which I will discuss about the applications in detail in my next session. But we will take up, because there are numerous applications where this CRISPR-Cas9 has been applied. So I thought of taking it as a separate session only. But again, because of its higher specificity, because of its very comparatively lower cost, using routine procedures, that how it can be done, and based upon how precise it edits, it becomes a very the need of choice, basically the editing mechanism. So there are many advantages, as I said. The gene repair is very precise, which is there. Then again, there are different mechanisms that can be used for therapeutic things. The advantage is that the target, the multiple targets can be targeted. The target specificity is also there. The CRISPR-Cas9 generation again is not a very time consuming or a highly costly mechanism. It can be introduced and created at a lower cost. So many advantages are there listed here, which make CRISPR-Cas9 genome editing system the choice system which is there. However, it does contain certain limitations. It is again not very 100% foolproof for off edits, off target edits. So it also has its own challenges. The basic limitation is the requirement of the PAM. Then another problem is the large protein complex that needs to be introduced into the cell. So if there is a heterologous expression that is occurring, that is a different thing. But if you want to create this molecule in vitro and then introduce this molecule into a cell, then Cas9 protein is quite large. Therefore, there can be certain kind of toxicity. The delivery mechanism is a little difficult, which is there. The non-specific edits could be there, although very low, but still it can be there. Then sometimes the homology that we define is not enough for these CRISPR-Cas nucleases to bind to those regions, and then PAM sequence is there. The specificity of PAM again is very limitation for the CRISPR-Cas9 system. So there are many advantages, but as I said, when you compare the different nucleases, genome editing tools, then CRISPR-Cas9 comes as a very handy method. Now we are having a lot of variants or orthologs of these Cas9 that combats with the large protein size thing. Again, we create very specific gene editing tools, which is causing the specificity to improve, the efficiency of the system to improve. So day by day this system has been improved for efficiency and specificity and ease of conducting the experiments, so that this is becoming the genome editing tool of choice. No wonder the zinc finger nucleases as well as talon nucleases are also used for genome editing. It has not just outdated them, but again it depends upon what kind of targeted that you want to achieve, what kind of facilities that you have, that you can choose to select this kind of systems. But as I said, because of its specificity and efficiency, a CRISPR-Cas9 system has become the, editing tool of choice. One important aspect in CRISPR-Cas9 system that I wanted to touch today, and which is a very important aspect, is the off-target genome editing, that activity that occurs. And then moreover, sometimes this CRISPR-Cas9 gene editing is required to be localized to specific cells or tissues at a specific time. So we do not want that any cell, or whenever we are targeting to a particular cell, then this spatio-temporal control needs to be there. What does this spatio-temporal control means? That the CRISPR-Cas9 gene editing tool, or the system that we have developed, it needs to be localized to specific cells. So spatially, if there are many cells, it needs to be targeted or localized to particular cells only or tissues only, and at particular time. So this CRISPR-Cas9, so there has to be certain kind of a regulation. I hope you understand what I am trying to say. That CRISPR-Cas9 system is a genome editing tool which, when introduced into a cell, will cause the double-stranded breaks as described in the mechanism of action. But now, how to control the action of these CRISPR-Cas9 molecule systems? Because we need to regulate it. One, because we don't want that it just goes and localize in all the cells which are present there, or in all the cells of the tissues that we want to edit. We might need only certain cells to be edited and not all the cells. And secondly, we also want that it is edited at a particular time, and it is not just edited at any time of, because there is a developmental plan. There is a process that is ongoing. The pathways can be activated or inactivated at different times. So we want a certain kind of a regulation. And as I said, the genotoxicity and off-target activity, we want to minimize that, so that the therapeutic efficacy, for example, is increased. And for these reasons, we need to have a spatio-temporal control of CRISPR-Cas9 gene editing. There are many mechanisms that has been described or used for this spatio-temporal mechanism. This is a paper from Nature, where they say there can be some physical control that can be put up. So there can be various kinds of magnetic activation or light source activations. All these kind of things can occur. That CRISPR-Cas9 system, when a certain wavelength of light is induced, then it becomes activated. Certain kind of magnetic properties are there that it becomes activated. Then certain chemical mechanisms can also be used. For example, certain enzymes or certain hormones or certain chemicals are used basically to modulate the activity of CRISPR-Cas9 system. So all these mechanisms which are shown here, they have their own limitations, because light, for example, it is difficult for the light or the photons to get introduced into certain cells or certain tissues. It will not penetrate those tissues, so that you cannot have this kind of a control. Similarly, for for example, these small inhibitory molecules. So these small inhibitory molecules may not even get introduced into the cells, or they may not bind at a particular site where the regulation or the control can occur. But I want to bring this concept here, because whatever tool you develop, because we want to harness it based upon our own needs, then how efficient that mechanism is. We must have mechanisms in place to control that system, to control that tool, and therefore I wanted to emphasize this fact. So in the CRISPR-Cas9 system, there are multiple mechanisms, the chemical means, the physical means, and the other different kinds of mechanism where they can be cleaved, or where they can be remain inactivated and activated at certain times, or activated in certain cells only. So they can be homology targeting, reaching at particular cells only. So there can be cell specific targets as well that can be introduced, but there has to be a control mechanism that controls the CRISPR-Cas9 system. So this CRISPR-Cas9 system is a very powerful tool. These arrays are present in the genomic DNA of prokaryotes, just to summarize what I discussed today. So these are present in the prokaryotes as a defense mechanism against exogenous DNAs. So they contain these spacers as the memory in between these CRISPR RNA arrays, and this CRISPR RNA arrays basically then get transcribed into cRNAs that can then be cleaved into discrete units containing these part of the spacer molecules, which will become the guide RNAs, and these guide RNAs will be homologous to the target sequences. Of course, adjacent to the PAM motifs. So that is the limitation of this CRISPR-Cas9 system. So when this guide RNA are cleaved into discrete units by tracer RNAs, it get associated with Cas9 molecules. It guides these Cas9 proteins to the target size, where the guide RNA has the homology to the target size, and creates a double stranded break that can be repaired. So this CRISPR-Cas9 system, what is its structure, how does it act, and what kind of controls are present so that we can modulate the activity of this CRISPR-Cas9 system is what we have discussed today. So I'll come back with another session in detail talking about the application of this CRISPR-Cas9 system. So we will discuss more about it. Thank you.